Welcome to our first virtual public talk series. We are now online for the Royal Astronomical Society in 2020, which will continue to May 2021. Hello, I am Lucinda Offer, Education Outreach and Bicentenary Officer. Our organization advocates for astronomy, geophysics, and space science in the UK since 1820. I am honored to start off our public talk series with you today and to introduce you to our guest speaker, Anna Nash, who will speak with us about the ExoMars rover PanCam and Planetary Protection. She is an AIT, which stands for Assembly, Integration, and Testing, and Contamination Control Engineer at the Mullard Space Science Laboratory, the home of UCL's Department for Space and Climate Physics. Having graduated from the University of Exeter, with a degree in physics, she wanted to gain more hands-on experience in clean room laboratories and was intrigued by MSSL's flight programs. Since joining MSSL, she has primarily been involved in the ExoMars PanCam project, the panoramic camera for ESA's Rosalind Franklin rover, which is now being launched in September of 2022. She has been involved in carrying out thermal vacuum testing and calibration of PanCam and helps to ensure compliance with the project's planetary protection requirement. A quick reminder that there will be a Q&A after the talk, so please get your questions ready. There are places to post your questions for our speaker if you've joined us directly on Zoom in the Q&A box, and I've seen some of you have already started to use it. Thank you very much. And we're also live on YouTube, so you're welcome to leave us questions there in the chat box. So please welcome our guests and thank you, Anna Nash, for being with us today. To start us off on our first Royal Astronomical Society public talk for the 2020-2021 season. On to you.
Well, good afternoon. It's a real pleasure to be with you all here today, albeit virtually, for, as Lucinda said, the first of the RAS public lectures for this season. A big thank you as well to Mandy Bailey for inviting me to give this talk in the first place. It's obviously a shame that we're not all together meeting in London, but a big thank you to Lucinda for moving this all online and making sure things run smoothly. So I'll just start sharing my screen. Right, so my name's Anna Nash, and I'm going to be talking to you a bit about planetary protection and the Rosalind Franklin Mars rover. But before we get onto the subject of planetary protection, I'll spend a bit of time giving you some background on the different phases of building a scientific flight instrument, which I hope will be of interest to you. So I work at the Mullard Space Science Laboratory, which is the home of UCL's Department for Space and Climate Physics. And this department is quite unique in that it's active both in academia as well as in bespoke space instrument engineering. And this special combination allows for physicists and engineers to work together to propose, design, build, and test scientific instruments for space and Earth observation. So I'm an AIT and Contamination Control Engineer. AIT stands for Assembly, Integration and Testing. So I, along with the other AIT engineers, am responsible for overseeing all the work that goes on in our clean room and seeing a piece of hardware from manufacture of individual components through to delivery and integration to the spacecraft. So that includes maintaining the clean rooms themselves, along with all the equipment they contain, operating our thermal vacuum test chambers, as well as being responsible for all the cleanliness of the flight hardware that we produce and test at MSSL, the importance of which I'll come on to a bit later. So MSSL has been involved in one way or another in 74 space missions, right the way back from Ariel 1 here in back in the 60s. You can see a picture here. Through to Cluster 2 in 2000. I won't mention Cluster 1. You can Google that if you don't know. And Swift in 2004, right up to More recently, Solar Orbiter, which was launched back in February of this year from Cape Canaveral in Florida. And luckily, I was able to make it out there to see the launch before we all ended up in lockdown, which was pretty spectacular to see. But the project that I've been most involved in, and which I'll spend the remainder of my time talking about, is ExoMars 2022, which consists of the Rosalind Franklin Mars rover, along with its landing platform, which is due for launch in summer 2022, pending further successful parachute tests, among other things, arriving at Mars in around June 2023. And it will be looking for signs of water-rich minerals, which are indicators of past life on Mars. It will be able to drill down into the surface to a depth of 2 meters, which is deeper than any previous rover. And it's thought that signs of life that may have been preserved in the first meter or so of Martian regolith may have been destroyed by radiation, because there are such high levels of radiation on the surface of Mars. But that perhaps below this depth, life may have been preserved. So MSSL's role in this mission was to design a panoramic camera system that would be the eyes for the rover. PanCam, as it is called, consists of 2 multispectral wide-angle cameras at either end of the optical bench. That's what we call this sort of white box that houses the cameras. And each of those uses a filter wheel with 11 different positions, including geological and atmospheric filters, to determine the composition of the geological formations on the surface of Mars. Now, they're stereoscopic cameras, so the images from each whack can be overlaid to create a 3D panoramic image. And in between them sits the high resolution camera, or HRC, which has a variable focus, which allows it to take very close, high resolution images of the local geology, as well as taking images further away. And this will allow the rover to plan its route, to avoid any obstacles, and to reach its target location as efficiently as possible. A mission such as this, where we're sending hardware to another planet, and one which experiences temperature fluctuations from about 20 degrees Celsius during the day down to about minus 120 at night, pose some serious technical engineering challenges, both mechanically and electronically. So first of all, the optical bench has to be as lightweight as possible, since every kilo of mass added means a more expensive launch, as more fuel and thruster power will be required. But it also had to be a very rigid structure, able to withstand the thermal contraction and expansion experienced over that temperature range. So the optical bench itself was machined out of a single block of aluminium using a technique called electrical discharge machining, that you can see here. And this technique can very accurately create the desired profile, and it was able to achieve a wall thickness of just 0.8 millimeters. So the optical bench itself is really very light. Another important requirement for the optical bench is that it must be light tight. So the lid has a radio frequency seal to prevent any interference. And the inside of the optical bench is also painted with a special space qualified black paint you can see imaged here, which has high thermal and optical absorptivity properties to suppress stray light. And since temperatures will generally be below 0 most of the time, the optical bench itself had to be thermally isolated from the rover using insulating standoffs. And the electronics inside had to then be thermally isolated from the OB in the same way. And this allows the electronics boards to warm up and operate at their optimal temperature without heat being drawn away by the rest of the rover. The outside of the optical bench was also painted in a white passive heat rejection coating to prevent any overheating as well. So MSSL also designed and made 2 of the circuit boards for the camera, the PanCam interface unit, or PIU, and the DC to DC converter. So producing electronics robust enough to withstand the thermal stresses and radiation exposure experienced by the rover was a really big challenge. And the electronics group at MSSL had to qualify every component and every solder joint to ensure that ceramic components wouldn't crack and solder joints wouldn't fail over the thermal cycle. You can see here one of the circuit boards, the PIU. And just to give you an insight into the intricacies of it, all 256 legs on this FPGA on this board had to be tinned and soldered by hand, with the soldering method having had to be qualified through testing and inspection under microscope of each of the solder joints on each of those legs. So it was really very involved. So for any flight instrument, there's always a number of models which are made for various purposes before the one that will actually end up on the spacecraft. For PanCam, first there was the structural thermal model, which is just the basic chassis of the instrument to make sure that it can withstand the thermal contraction and expansion. And then the engineering model is made to slightly more exacting standards with any modifications that have arisen from the STM. And it also includes the electronics boards inside, which will allow for functional testing of those circuit boards. And then because this is a relatively long-term mission on a planet that will experience a wide range of temperatures, a life test model was made. The LTM was subjected to vibration testing to simulate launch, shock testing to simulate staging of the rocket, and then it was thermally cycled 300 times to simulate the 300 or so sols that it will be in operation for on the surface of Mars. And this was a really vital stage of testing, as some of the components did fail during these tests and required redesign. And after this came the proto-flight model, which I've spent much of my time at the lab working on, and which was completed about 1.5 years ago and is now integrated on the Rosalind Franklin itself. So then after the PFM comes the FS, or flight spare, which is finished and is currently sitting in one of our clean rooms for safekeeping. The flight spare is made as a potential swap if anything were to happen to the flight model before launch. But more often than not, it isn't launched and is instead used for ground-based tests and simulations in case any issues arise with the instrument once it's in space. Flight spares also sometimes get repurposed for other, perhaps similar missions. So for the proto-flight model and the flight spare model, once individual base components of the instruments are manufactured, a piece part solvent clean is carried out, and then each part is baked out in a vacuum chamber, which is all done for the sake of cleanliness. Particular and molecular cleanliness is a significant requirement for any flight project, which I'll come to in a minute. So once this is done, the parts of the instrument can be assembled. And then a whole series of tests are performed to ensure that the instrument functions the way it should. So in between each stage of assembly, integration, and testing, visual inspections of the hardware are done under UV and white light to monitor the cleanliness of the instrument as it completes each development phase. which leads me onto one of the major challenges of all flight programs, and that is keeping the flight hardware clean. Now, in any other profession except perhaps medicine, cleanliness might seem fairly inconsequential, although perhaps not anymore in our post-COVID world. But I have to say, when I started this job, the word initially gave me flashbacks of my mum telling me off for the state of my bedroom. But for any hardware that goes into space, cleanliness is really very important. So one of the big reasons for this is down to the process of material outgassing, which is the release of gaseous molecules that have previously been trapped or absorbed in the material. Now, pretty much every material goes through this process, and you can see it in everyday life because certain materials often have a smell associated with them. For example, rubber has quite a distinctive and pungent smell, and so it follows that it outgasses quite a lot. The same goes for the materials that make up the internals of a car. That distinctive new car smell is again those plastics and polymers outgassing. Those are just molecules that are being released from the material and then are coming into contact with the odor receptors in your nose. So non-metallic materials, particularly organic compounds, typically outgas more than metallic materials. And as the temperature of a material increases, its rate of outgassing also increases. Eventually, though, the outgassing rate will drop once the bulk of the volatile molecules from the material have been released. So if materials haven't had the chance to outgas enough before being launched into space, they will once they're there. Those molecules are then released and then condense on any cold surfaces, which on a space instrument are often the sensitive optical surfaces. And this can cause a massive problem as the functionality of the whole instrument is affected. This actually happened in the case of NASA's Cassini mission back in 2001, where a haze was detected on images captured by their narrow angle camera due to contamination or the lens or detector as a result of outgassing. Fortunately, the NASA team were able to remove this haze by taking the camera through 4 decontamination cycles, whereby it was heated up to bake off that contamination. So materials used for space instruments are carefully selected to be those that have low outgassing levels to begin with. And then each part, down to every nut and bolt, is baked out before and after assembly to release those molecules and reduce the likelihood of a contamination event once it's in space. So we categorize contamination into 3 main groups: particulate, molecular, and biological. Most flight projects only tend to have requirements for particulate and molecular contamination, and the level of cleanliness also varies depending on what type of instrument it is and where it's headed to after launch. For example, for sensitive optical systems like focal plane arrays or mirrors for space telescopes, such as that for the James Webb Space Telescope you can see here, cleanliness requirements are very stringent. As I mentioned earlier, for any piece of hardware, for any project, cleanliness is also recorded at each stage of assembly, integration, and testing by inspection under UV and white light. And we record that down to every particle or fiber, and it takes rather a long time. So we also monitor whatever environment the flight hardware is exposed to using PFO or particle fallout and MOC or molecular contamination witness samples, which are just representative of a given surface on the flight hardware. So a PFO is a smooth glass plate that collects particulate contamination, which can then be measured using a photometer. And a mock sample is an infrared transparent glass window which collects molecular contamination. And this can then be measured using Fourier transform infrared spectroscopy. From these results, you can then infer what contamination the hardware may have collected. So during vacuum chamber tests, molecular contamination within the chamber is also monitored using a thermoelectric quartz crystal microbalance and a residual gas analyzer. So a TQCM is a set of two very thin quartz crystal discs in which oscillations are induced at a given frequency when they're subjected to an alternating current, which is an example of the piezoelectric effect, if you're familiar with that. One crystal is a reference, which is enclosed within the TQCM, so its frequency remains constant. The other crystal is exposed to the chamber environment, and it's kept at -20 degrees Celsius, so that it attracts molecular contamination within the chamber. The reference crystal is kept at around 1500 Hertz, higher than the base frequency of the exposed crystal. And so as the exposed crystal collects this contamination within the chamber, the increase in mass will cause its frequency to drop, increasing the difference between the two frequencies. This is represented by the red line on the graph. This graph is actually from one of PanCam's thermal vacuum tests. It's impossible to completely stop outgassing, but eventually when this red line reaches less than 1% deviation from linearity, and the rate of outgassing represented by that black line levels off, the vacuum bakeout is finished. So the TQCM tells you quite accurately how much contamination is in a chamber, and so how much contamination the hardware is seeing. An RGA is a residual gas analyzer, which is a quadrupole mass spectrometer with a tungsten filament which ionizes the atoms and molecules within the chamber and detects their mass. The most recent RGA models can detect down to 10 to the -14 millibar partial pressures, which is really very sensitive. So the TQCM and the RGA work in tandem to provide us with real-time data telling us how much contamination is in a chamber and what that contamination is made up of. So as you can see, ensuring that flight hardware is clean for any given project is quite involved. But the ExoMars PanCam instrument has been even more complex in terms of cleanliness. As well as having all the usual cleanliness requirements of a flight project, we also had to fulfill the planetary protection requirement. Now, planetary protection is an international standard established by the Committee on Space Research in compliance with the UN Space Treaty of 1967 to protect against interplanetary biological and organic contamination. So a key consideration in the exploration of other planets like Mars is that we don't accidentally introduce Earth-based biology. This could have an adverse effect on the planet's environment and could also jeopardize the scientific investigation, the very reason that the Rosalind Franklin is being sent in the first place. And for missions where a return journey is planned, perhaps to bring back samples, which isn't the case for this mission, there are also guidelines on how to do that safely to protect the Earth from extraterrestrial contamination. So given the planetary protection requirement of this mission, a lot of research was conducted to try and understand the enormity of the task. So just to give you a sense of it, in 1 gram of soil that you might find in your garden, for instance, there are around 25 billion CFUs. A CFU is a colony-forming unit, which is another way of saying a viable bacteria or fungal cell. And now that I'm sure you've all had your lunch, I can tell you that on just 1 centimetre squared of skin, say on the tip of your finger, there will be around 10,000 CFUs living on it. And in just 1 metre cubed of ambient air, there will be around 2,000 CFUs. So let's compare this to the PanCam requirements. So over all external surfaces of the PanCam optical bench, we weren't allowed more than 100 CFUs. And over all internal surfaces of the OB, we can have more than 1,000 CFUs per metre squared. I should also state that planetary protection is in place for the entire rover and landing module, of which we've only built one part. So Airbus and the other organisations who are producing other parts of the rover will also have to adhere to the same standard, though their CFU budget will vary from subassembly to subassembly. So as well as monitoring particulate and molecular contamination, as we do with other missions, we're also concerned with bioburden reduction and monitoring. So we've developed several measures to reduce bioburden in our ISO 5 clean room and on flight components, as well as working practices to ensure that bioburden isn't introduced into the clean room and transferred onto the optical bench. So firstly, the way we enter and operate in the clean room is different to other clean rooms. So we only use sterile clean room garments, and we have to double glove using sterile nitrile gloves. And when you're working in a clean room with planetary protection measures, you have to keep track of every single thing you touch. Even if there is a slight possibility that you've touched something that isn't sterile, you have to put on a new pair of gloves before touching anything that is, especially any flight hardware. So bioburden transfer is extremely easy to do. And unfortunately for us, the 5-second rule does not apply. We also regularly sterilize the workstations using hydrogen peroxide, and we're only allowed to use chemicals that are 0.2 micron filtered, as this is the average diameter of most bacteria. As well as this, we try to handle the flight hardware as little as possible to further reduce the risk of bioburden transfer. So in the ISO 5 clean room, there is a horizontal airflow from one end of the room to another, which drives out any particulate contamination. So we also try to ensure that we work downstream of the flight hardware, so that there isn't the risk of particles which could contain bacteria falling off of us and blowing onto the flight hardware. We also employ a 3-person buddy system, which you can sort of see imaged here, where one person is the designated clean buddy, and they will actually handle and work on the hardware. The second person is the dirty buddy, who will assist without direct contact to the hardware. And the other person, it just observes to make sure that no one accidentally transfers bioburden or makes a misstep, which is very easy to do. So a clean room under these planetary protection measures is actually cleaner than an operating theatre. And in addition to clean room working practices, we also process all flight hardware components and support equipment into the clean room with additional measures. So for most hardware components, after it's gone through 7 stages of solvent cleaning, it is then heated up to above 110 degrees Celsius in our DHMR oven, which stands for dry heat microbial reduction. And this is to kill any bacteria which have survived the cleaning process. The duration of heating depends on the temperature to which you can take the material, as well as the type of material it is. For example, at 118 degrees Celsius, surfaces that are free for gas exchange, say just a bare aluminium plate, will be required to be DHMR'd for just under 2 days. But mated surfaces, like two surfaces bolted together, will have to be DHMR'd for just over 3.5 days. And materials which classify as having encapsulated bioburden, such as epoxies, O-rings and coatings, will have to be DHMR'd for about 5.5 days. So as you can imagine, just getting all the components ready for assembly takes some serious planning. We also use an autoclave, which uses steam at high pressure to sterilize anything that's placed into it within an hour. And while most flight hardware components are not compatible with this method of sterilization due to potential water uptake and damage, it has been an invaluable piece of equipment used for verifying bioburden reduction. And verification is the all-important thing. We need proof that we're fulfilling the planetary protection policy. So at each stage of the assembly, integration and testing, biological assays were taken of the hardware. Now, an assay involves wiping the surface of the hardware using a sterile swab, not too dissimilar from a COVID-19 test swab, if you've ever had the misfortune of getting up close and personal with one. We wet this with sterile water, and then these swabs are then sealed in a tube, which have been sterilized by the autoclave and are then sent off for incubation and are checked for the growth of bacterial colonies every day for 3 days. So this first image on the left here shows pretty heavy bacterial growth on an isolation plate. And I'm pleased to say that this is not a PanCam sample and is just there for comparison. The middle photo is a PanCam isolation plate, and it has 3 CFUs, if you can see them there. But even a CFU count as low as 3 isn't good enough. And we're required to re-clean and re-assay any surfaces that come back with a CFU count higher than 1. So what we're really aiming for is a 0 CFU count, as you can see in this last photo. And with the measures that we put in place, most of our samples successfully come back with 0 counts. And so, as I said, assays are performed throughout the whole AIT process, but perhaps the most crucial ones are the ones taken pre-delivery. So without acceptable bioburden levels, we wouldn't be able to deliver PanCam. And unfortunately, we weren't able to do an end stage sterilization, as not every component within PanCam is suitable for DHMR. So these assays were performed on the PFM-OB on the 13th of May in the locations you can see in this diagram in orange. And I can happily say that for both the internal and external surfaces, we were within the requirements set by the project, with a total of 50 CFUs present on external surfaces and 364 CFUs per meter squared on internal surfaces. So having succeeded on all these different engineering fronts, PanCam was delivered to Airbus in Stevenage on the 20th of May 2019, and was integrated onto the Rosalind Franklin in August 2019. So the whole rover as an entire assembly has undergone environmental testing, a few modifications, and general operations maintenance is now taking place at Thales Alenia Space in Turin. As many as you may or may not have heard, a launch was scheduled for a couple of months ago, but has now been postponed to 2022 to allow sufficient time to complete all the required testing, as well as to account for the challenges to the schedule brought on by the COVID-19 pandemic. So while this delay does unfortunately mean having to wait another 2 years for launch, I hope you'll agree that an unsuccessful launch or landing on Mars isn't really worth the risk when so many people in so many countries have all put in that work that's led us to this point. So we patiently but eagerly await launch in 2022. And now it's worth saying that scientific and engineering endeavours are never just the work of one person. So to give you an opportunity to have a bit of an overview and a recap, as well as an opportunity to hear from some of the other scientists and engineers involved in the PanCam project at MSSL, here's a short video about PanCam.
PanCam is a panoramic camera. That's what it stands for.
PanCam, this is the scientific eyes of the Rosalind Franklin rover. So we're looking at the scene on Mars, trying to do some geology, trying to do some atmospheric science.
The instrument is a fairly plain white box, essentially, but it's what's inside the box that's interesting.
We've packed as much science as we possibly can into what is basically a camera system.
We have 2 wide field cameras on the ends.
Stereo imaging cameras of the wax, one at each end. So on a stereo baseline of about 500 millimeters for stereo imaging and plotting of routes on the Martian surface. So looking at what's in front of the rover and working out where it can go.
So each of these wide-angle cameras has a filter wheel in front of it with 11 positions, and we can move that around to look at the different colours of the light coming from the surface of Mars. And so with that, we're able to not only build up a colour picture, but also to look at composition of the rocks, in particular looking at the light which is reflected from the rocks, to look at the minerals in them, and to look particularly for water-rich minerals, because of course we're looking for water and life on Mars.
And then we have a high resolution camera.
So the HRC light path comes in, goes, hits a fold mirror, goes through a focusing mechanism in this area, and then projects onto a sensor down at this end.
This will enable us to actually see fine-scale, small-scale details on the Martian surface. For example, texture on rocks, or it will enable us to see single grains on the soil, and that's very important for the geological investigation of the surface.
We were PIs on the optical bench, and we produced the PIU and DC-DC converter, and they were built here.
We already delivered the hardware, so it is now in still finish in Airbus. Now they're putting all the things on the rover. The project starts since 2004. So what you're talking about is 15 years of our life is working on it.
I remember the first time I saw part of PanCam and then got to handle some of it, and it was a pretty amazing experience to think that, you know, things that you've touched and helped with will hopefully be on Mars soon.
It would be a fantastic discovery to actually be able to find evidence for life anywhere else on the universe, and Mars is the nearest place we can look, and so our rover mission is specifically designed to do that.
Brilliant. Well, thank you very much for tuning in and listening. It's been a real treat to be able to give this talk virtually in the absence of being able to meet together physically. So I believe there's now a few minutes for some Q&A if anyone has anything to ask. Thank you very much.
Anna, thank you so much. What an interesting and informative talk. It was really great. You gave us lots of detail and lots of information to know and understand about what it really takes to get a rover to Mars. And that was brilliant. Thank you so much for being with us. So we do have some questions in the queue, and we'll just take a few seconds here to remind everybody that we will be doing questions and answers now. So if you have a moment and you haven't yet taken one, go ahead and add it to the chat box at YouTube where we're live streaming, or in Zoom if you're with us and you happen to register via Eventbrite and be with us directly on Zoom. We do have a Q&A. We have a queue actually adding up right now for you, Anna. So we've got plenty of questions there that will get us started, but we've got about 15 minutes to take questions. So without any further ado, let's get started. So the first question we have, they say, a guy, he says, hi, Anna. How would MSR, for example, avoid backward contamination of samples to Earth? Is this done on the Martian surface or an orbital transit back to Earth? Thanks.
So honestly, I'm not entirely sure. So this isn't back contamination back to Earth isn't something we've considered for this project. I would imagine that it's partly the way that the samples are received back on Earth. So sort of doing it in a very controlled way and holding those in sort of quarantine to analyze them. But yeah, I have to say, I'm not entirely sure. I think it would depend as well on the type of samples you are bringing back and where you were bringing them back from. So yeah, sorry, I can't give a more detailed answer to that.
Oh, that's okay. We can't, you know, as scientists, and I've done about 14 of these talks this year already through this pandemic, you know, and I've learned firsthand that we don't know everything, even though we're scientists and we're in the thick of it, research every day. Some of us focus on one or a few things, so we can't know it all. So we definitely appreciate whatever you can let us know about. And who would have thought that we would have so much in common with the ExoMars rover, with this pandemic and contamination and assay swabs. That was really great that you put that in there and have been very you know, present with what we're going through right now. So this person asked, don't swabs leave fibers and residues?
Yes, so the swabs that we would use are, yeah, they do leave a certain amount of fibers. Fortunately, it's not very many, and so every time we would perform biological assays directly after, we would then inspect where we had just taken the samples, and we would remove any fibers that were left. Unfortunately, they fluoresce under UV light, so it's very easy to see them and then remove them. So yeah, that's all taken into account. Yeah.
Okay, this speaker asks, isn't even a count of 100 biologically high inane area where life could have been or could now thrive?
So I think basically it's impossible for us to produce hardware where there would be zero counts completely. And so there is a certain amount that is allowable. So things like in the worst case scenario, if it were to crash on the surface, it is a small enough number that they're reasonably confident that it wouldn't produce adverse effects. So that's why sort of the externals are have higher or tighter restrictions than the internals, as it's more likely that just the externals will come into contact with the surface. But yeah, it's partly that it's the practical nature of it. You'd have to do it, all the assembly and everything sort of robotically, I would imagine, to completely eliminate biological contamination. But yeah, if we know what the biological contamination levels are, that can be taken into account when actual samples are being taken on the surface, to make sure that, you know, what we're picking up isn't stuff that we've brought with us, but that is actually there. So yeah, it's all taken into account when data is collected.
Great. So Patrick asks, great talk, very interesting and informative. And this is something also that we've been doing with the pandemic. Some people who are sterilizing objects, they're using UV light, for instance. And Patrick asks, can radiation be used to sterilize equipment?
Yes. So a lot of our equipment and sort of consumable items, so like our garments and our gloves, are irradiated, so that we're confident that they're sterile. This can't always be used for sensitive equipment though, as well as it being quite sort of using UV at those sort of high intensities is quite dangerous and needs to be done in a very controlled environment, and those facilities aren't necessarily available to us. But yeah, I think more likely is that you can get sort of UV degradation of the materials from high intensity UV light, which is obviously something we're really trying to protect against. So yeah, that's kind of a big reason not to use that for the actual flight hardware.
And that makes a lot of sense, especially on its flight to Mars, and when it's on the surface, it's going to be exposed to other, lots of radiation there. So that's a good point there. This is a really interesting question, I think, because I'm curious about this too. John asks, how will the equipment be kept clean if there is a 2-year delay to launch?
Yes. So if you, so for example, we're keeping the flight spare in one of our clean rooms, which is the actual flight spare is biologically clean. So it's kept inside a container, which itself has been sterilized. And then essentially, if you're not entering the clean room often, and if you're not touching any of the equipment or the hardware, then you can be reasonably confident that it's not gonna kind of accrue spores or biological cells, but it is something that has to be taken into account, and storage facilities have to be sort of verified that they're sterile. When it comes to the whole rover itself, I don't know how they manage it. Obviously, something that big and complex is significantly more of a challenge than just the PanCam that we have to deal with. But yeah, but assays again will be taken nearer the sort of launch time. So yeah, it will be monitored constantly.
Okay, excellent. Still, we have lots of questions here. Apart from travel, does COVID affect this less, given the clean suits ought to keep everyone COVID safe, and so social distancing in a clean room should not matter? So do you guys have to still social distance even though you're in a clean room?
We try to as much as possible. We have ourselves, we have limits on the number of people that are allowed into a clean room. We have that anyway, but it's become even more stringent during COVID. But you're right. I mean, it's a pretty safe place, as we wear masks and suits and gloves. So you are well protected. So yeah, it's meant that we were able to come back to the lab in July, able to get back working. So yeah, it is a perk of all the PPE we have to wear on a daily basis.
So here's a kind of a similar question. Asks, if a sample test comes back with 0 CFUs, is there a chance that CFUs could form before the mission launches?
Well, they couldn't form, I guess, spontaneously. They would have to be transferred onto it, which is why we do regular sampling. And so it would be sampled with us here before we deliver it. And then it would be surfaces on the rover would be sampled once it's been integrated. Obviously, that's now down to Airbus and Thales Alenia. But they wouldn't just sort of pop up. But yeah, transfer is very easy. And so yeah, you gotta keep a close eye on it.
Paul asks, now that, I don't know, maybe since you've sent the PanCam back in 2019, what other projects are you involved with? Are you onto something different now? Yeah,
so we've moved pretty quickly onto the next project. The big one here at the lab at the moment is called Plato. It stands for Planetary Transits and Oscillations. So it will be it's a space telescope looking for exoplanets. And so we're involved in building the electronics for the cameras on board that space telescope. So yeah, it's a big project for us, and so yeah, we're busy cracking on with that at the moment.
So Julia was paying attention to your presentation. She noticed there was a spike in the outgassing, but she didn't say specifically what that might have been from your presentation. So she wants to know, but do you know what was released, or is it a combination of materials and molecules?
So it's usually a bit of a combination. But so yeah, what we often see is peaks for water and for hydrogen, nitrogen, so the sort of things that we would expect to be in a chamber. And we monitor those levels throughout the test. And yeah, so I think in this case, there would have been a bit of a spike. Sometimes just the way things go, you might get a pocket of gas or something that needs time to be released in the vacuum chamber, and so you might get that. Also, another piece of equipment inside the vacuum chamber is something called a scavenger. So it's basically just a block of metal that is cooled down to minus 180 degrees Celsius using liquid nitrogen. And this is basically just to grab all of the molecules that are floating about in the chamber that haven't been sucked out by the vacuum pumps. And so, but if the scavenger fluctuates a bit in temperature, if it warms up a little bit, it will release some of those molecules, and just that tiny amount will cause a bit of a spike in the TQCM. So we try really hard to keep the scavenger at a steady temperature, but occasionally if you get a blip, then it will cause, you'll see it on the TQCM and the RGA. But yeah, hope that answers that.
Yeah, no, that's great. We have quite a list of questions already, and only about 6 minutes more. Let's see how many more we can get through them. AC asks, any attempt in lifetime testing of the camera to attempt simulated particle accelerator radiation exposure to model space and Mars surface conditions?
Not that I'm aware of. Yeah, so the life test model, yeah, really just to make sure that sort of the mechanical integrity of the parts, each of the parts that make up PanCam wouldn't fail during those temperature fluctuations. In terms of yeah, I think that's the only sort of radiation sort of component that was taken into account. Yeah, other than that, I don't think so.
You might have already asked this, and maybe some of it you don't know, but John wants to know, how is the contamination security of the PanCam and rest of the hardware maintained during the 2 years prior to launch? You might have already touched on that a bit.
Yeah, I mean, it's really down to storage, and we're we're always sort of given advice and support from ESA, and it will be the same case for the other organizations involved. So we kind of check with them and make sure that the way we're storing it is approved. And yeah, it's just not touching it too much, and yeah, making sure that it's all safe and secure, and just yeah, gotta leave it for 2 years, and then obviously check for biological contamination just before delivery or launch. But yeah, bit of a wait and see game.
Yeah, that's kind of a scary thing, like waiting for a couple of years and then testing it right before launch, and then if it is contaminated, well, they have to take it apart and clean it up, and that sounds like a huge ordeal.
Yeah.
Yeah. Okay, so David asks, how do the precautions this mission has taken to prevent planetary infection compared with manned missions? So not sure if you would know this yet. The Apollo, so he says, for example, the Apollo lunar missions, and it strikes him that a human body carries a lot of CFUs.
Yes. So, I mean, with the Apollo missions, there was not really a sort of an investigation into whether there would be life on the moon. So it wasn't a consideration in that sense. Obviously, they weren't worried about, getting false positives. But yeah, so it depends a lot on the scientific mission, and whether scientists think it's likely that there would be life there that could be adversely affected by humans. So yeah, I'm not entirely sure, and I'm excited to sort of find out how they approach this with sort of the Artemis mission, NASA's Artemis mission, and kind of looking forward to sending humans to Mars. How that's all navigated, it'll be really interesting to find out.
Yeah, probably a much bigger ordeal. We've done over 20 years, probably actually longer, since IBMP in Russia. They've done lots of hazard kind of biomedical research on humans, space exploration. So we do have several decades of data. It's just, yeah, it's a huge, big risk and leap forward with humans. Okay, so we have a couple really interesting questions from an anonymous person who, it's a really great question because RAS often does GCSE talks, and for students, they want to know, like, well, do you have a master's, a PhD? How did you get your job at MSSL? Your AIT job sounds really cool. Is it a first degree in physics engineering? Is that enough for that role, or was there postgraduate qualifications that you had to do?
Yeah, so yeah, it all happened very fortuitously in a sense. Yeah, so I just have a physics degree at the moment. Yeah, and I essentially, when I left university, I wasn't entirely sure what I wanted to do. I knew I was interested in space, but I didn't know entirely how sort of to get into it. And I saw a job at MSSL for working in the clean rooms, and I'd worked in a clean room for a couple of months for a project at university, and so I thought I'd give it a shot. And yeah, so I started here, and I've learned absolute heaps. Yeah, so a postgraduate degree is not necessarily required. Obviously, it always helps, I guess. And it's I'm planning to do a master's in the next couple of years. But yeah, so a mechanical engineering degree, an electrical engineering degree, a physics degree, all really useful in different ways. Yeah, so our role here, as you might have been able to tell involves a lot of different sort of subject areas. So there's material science, there's mechanical and thermal engineering. You need to know a certain amount of electrical and electronic engineering concepts. So yeah, all of those subjects are really great to get into this sort of field.
Excellent, Anna. It has been an absolute joy having you with us. That's really great information. It's really important for young people to be able to get involved with all the future missions coming up. And so, exactly, you just get your foot in the door. You prove who you are as a human being with your colleagues. And if they like working with you, you get up for certain positions, or you go for them if things that you're interested in. Exactly. So I think that's fantastic. Great answers, great presentation. I really enjoyed it and appreciate your time with us. So thank you again for being with us for our public talk. If you have any last words, you're welcome to share them now.
I'll just thank you all for tuning in. And yeah, it's been a great afternoon for me. So thank you very much.
Well, it was a pleasure having you with us and a great kickoff to our Royal Astronomical Society's public talks. With that, I'm going to say goodbye to everyone. Have a great afternoon, and we'll see you next month.